Okay, continuing on with our lymphatic cells. So we have the category of phagocytes. So the first one we have is macrophages. So I nicknamed them the jocks because their job is just to eat stuff. So have you ever seen a football team and a giant pot of spaghetti disappear? Yeah, same thing. So our macrophages, basically any kind of debris or any foreign pathogen that it detects, it's going to just phagocytize it, break it all up, and then release all of the particles to be disposed of. So these macrophages can also work as antigen-presenting cells. So instead of breaking down the entire pathogen, they'll still have that little bit of the foreign antigen that they'll display on the cell to activate other immune system cells. The other type are the dendritic cells, which I call the nerds because they hang out in the first line of defense, or what I call the office. So they're still just like your regular macrophages. So they're going to phagocytize anything foreign that they come across. That can be found in the skin. So if you remember that from chapter 6. Also found in different mucous membranes. So their job is just to alert the immune system to pathogens. So because they're in that first line of defense, they're the first ones to come across any kind of foreign pathogen. And then lastly, our reticular cells are just helping to with structure in our different lymphatic tissues. Okay, let's look at these freshmen. So we have our never let monkeys eat bananas. So our white blood cells, just what they do in general. So the first ones are our neutrophils. I call them overeager because they will detect a foreign pathogen once they are attracted to that area of infection, and then they have multiple ways of just killing off stuff, so they just go after it. So for our neutrophils, they can kill by phagocytosis, so they can internally ingest the foreign pathogen, so eat its stuff. Or they can go through what's called a respiratory burst. So a respiratory burst is a cloud of bactericidal chemicals. So it just releases these chemicals like a cloud around it. Creates a killing zone around the neutrophil, but unfortunately, even though it's going to kill several bacteria around it, it's going to kill itself in the process. So with your neutrophils, when they die off as part of this process, this is why your mucus ends up green, because there is a green pigment in neutrophils. So that's one way of determining how sick you are. How many dead neutrophils do you have? So the greener your snot, the more, killing off of an infection you have. So for them to get to an area of infection, they are attracted by what's called chemotaxis. So those T helper cells, when they secrete those interleukins, those chemicals will create a gradient of chemicals. So as those chemicals go down their concentration gradient, they're going to create a gradient that attracts these neutrophils to that area of infection. So let's say if your thing is energy drinks. So if I were trying to, kidnap you, I could place out a increasing concentration of something that you like an energy drink or candy. And that increasing concentration of something that you like would eventually attract you to the white van, and then it's all over with. But that's what the neutrophils do. They're attracted to that area of playground. Sorry, I'm being real dark right now. So attracted to that area of infection, and they go on that killing spree. Next one, the lymphocytes. So these will differentiate into our natural killer cells, B cells, and T cells. So there are most of what our circulating blood contains are the T cells, and then some B cells and some natural killer cells. But most of them are our T cells. Okay, monocytes. I call them the JV sports because they're going to mature into those macrophages, those big eaters. So they're going to wander around in our particular connective tissue where it's loosely organized, and they can kind of migrate, and when something crosses that epithelial tissue, that tissue that is exposed to the outside environment, pathogen comes in, those wandering macrophages can then start eating up stuff. So phagocytosis. Some of our macrophages are fixed, so they're stuck in wherever they are located. So we have our microglia in the central nervous system, so they can't really go anywhere. The alveolar macrophages, so in the lungs, and then hepatic in the liver. Eosinophils. So, second to last one, I call this the weird kid. Their job is to fight off allergens or parasites. So, they're found in your mucous membrane, so something that is exposed to the outside environment. And their job is going to fight off foreign things that sometimes either can just irritate you or specific stuff. So again, the weird kid. So their job is to kill off parasites like tapeworms and roundworms. And they do it using different oxides that's going to break down their molecules, and eventually that's going to break up those, whatever it's targeting. So superoxides, hydrogen peroxides, that's why it bubbles as it's doing its job, because it's breaking down anything that is biological. And so when it breaks down that, it ends up breaking apart into hydrogen and oxygen, and that creates the bubbles. And then also has different toxic proteins to kill off stuff. They can also phagocytize antigen and antibody complexes. So when those antibodies bind to those foreign antigens, those eosinophils can target it and then destroy that foreign pathogen. And the last one, basophils. I call them the teacher's pet, basically because their job is just to be helpful to all of your other leukocytes. So they're going to secrete leukotrienes, histamine, and heparin, and that helps to either attract some new white blood cells, so neutrophils and eosinophils, to an infection. Histamine is a vasodilator, so that helps bring fluid towards that tissue area, which is going to bring white blood cells. It's going to bring antibodies to that area. And then lastly, heparin, which inhibits clot formation. So the clots in a blood vessel would prevent leukocytes from getting to that particular area of infection. So just being helpful. Okay, our last set of players are the journalism squad. So these are antimicrobial proteins. So we have two different types. We have our interferons and our complement proteins. So both of these are going to inhibit microbial reproduction, basically stop pathogens from multiplying inside your body. So the first one are interferons. I call them newspaper. They're the ones to give breaking news of an infection. So these interferons are secreted by the infected cell, particularly the ones that are infected by a virus. So it doesn't help the cell that actually has secreted these interferons, but the neighboring cells will know to form a defense against becoming infected. So it works by binding to receptors on the neighboring cells, and that's going to activate a process within the cells to start that defense system. So with the neighbor cells now being alerted, they will make different proteins that, again, there's our defense system, prevent viral genes from replicating inside their cells. Those interferons can also activate your natural killer cells and the macrophages to come and destroy that infected cell. So before it can release the viruses that it has produced when the virus has taken over its protein producing and genetic material producing like organelle. So it turns into a virus factory. So basically destroy the factory before the product can be released. It also activates the natural killer cells to basically just destroy the cell in general. Again, that's going to help to break down that cell and destroy it before it's able to fully become a factory or before it's able to release out those viruses. Next type of protein is a complement protein. I call this yearbook. This is both part of adaptive immunity and innate immunity. So they work in three different pathways. So there's classical, alternative, and the lectin pathway. The classical pathway is for adaptive immunity, so against a specific pathogen. So these proteins are going to form a antigen-antibody complex And that helps with bringing in other proteins that are going to help to target the pathogen and kill it off. Next pathway is alternative and lectin pathway. So this is innate immunity, so our natural born immunity, so very generic no matter what type of foreign pathogen. So with the first one, the antibody is specific against that specific pathogen. So these complement proteins in the alternative and lectin pathways, they're going to bind to the actual foreign antigen by itself. So it doesn't need an antibody to target them. So that's going to bring in other complement proteins in order to, again, kill off the foreign pathogen. So you don't have to know this chart, but I was just showing you how complex it is, where you have your different type of complement proteins and they cause other stuff to happen in the cell. So they'll cause inflammation, they'll cause immune clearance, which we'll talk about later, phagocytosis, as well as cytolysis, breaking up that cell. So here are the 4 ways or 4 methods of complement protein pathogen destruction. So ways that these complement proteins can help with killing off a pathogen. So the first one is inflammation. So that last slide I showed you, so these 4 here at the bottom. So first one's inflammation. So it stimulates histamines to be secreted out, and that attracts our neutrophils and macrophages. So get them to come and do their job, basically speeding up the whatever infection that's happened. So that's why sometimes if you have a cut or something happened to your body and it swells, it's because you have a bigger flow of those white blood cells there. So even if you've torn tissue, those macrophages still need to come in there and get rid of any kind of debris. So the next one is immune clearance, which I've kind of done a little cartoon here at the top. So this is a part of adaptive immunity. So we have our antibodies that have bound to the specific pathogen. Then this complement protein binding to our antibody foreign antigen complex, it creates a bridge between all of this and our red blood cell. So the red blood cell can then, kind of like a trailer hitch, take the pathogen that is stuck to the antibody and the complement protein to the liver and the spleen. So you're going to have a bunch of small blood vessels in those areas that can strip off all of this portion. So the complement protein, antibody, and the foreign pathogen. Red blood cell can go on its happy little merry way. And there, where you do have a bunch of white blood cells, they can go ahead and kill off that pathogen in that area. So this is the principal way that foreign pathogens are cleared out of the bloodstream. Third method of complement protein pathogen destruction is phagocytosis. So we already talked about this. So our macrophages will bind to foreign pathogens. Sometimes they go through what's called opsonization. So there are binding sites that are created on these foreign pathogens that these phagocytes can basically makes it a whole lot tastier. So it helps them with binding to that foreign pathogen and allow it to consume it. So break it apart into pieces where it can't do what it would normally do. So, cause an infection. And the last one is cytolysis. So we have these complement proteins. So you'll have one of your complement proteins will bind to the foreign pathogen, and then it'll recruit the rest of these. So part of that is this ring here will create a hole in the plasma membrane of that pathogen. And this creates what's called a membrane attack complex. That hole in the pathogen will eventually cause the cell of that pathogen, or just the package of that pathogen, to rupture. So it'll break apart and no longer able to be infectious. Okay, last piece for the generic function of the lymphatic system before we go back to immune function is lipid absorption. So we'll talk about this more in our digestive system chapter, but as your fats are broken down into smaller and smaller pieces, eventually you get to your triglycerides. Those triglycerides, they're packaged in their own little vehicle. Same thing with, for example, your cholesterol or any of your fat-soluble vitamins. They're all packaged together. And that package, again, too big to get into the capillaries of your bloodstream. So that larger package still is small enough that larger package is called a chylomicron, by the way. That package is small enough to get through the flaps in that lymphatic capillary and then just flow through your lymphatic system and eventually get dumped into the bloodstream, where then your body can make use of those lipids. Okay, I'm going to stop here and start up, because this is a whole nother section, doing our different types of immunity. So just kind of explaining them more. See ya.